Assalamu alaikum everyone. In this video lecture, we will be discussing February-March 2024 paper 2 variant 2 of AS level biology. Let's start. Question number 1. Part A. Figure 1.1 is a diagram representing part of the phospholipid bilayer of cell surface membrane. So over here we can see that this is the part of the phospholipid bilayer of cell surface membrane. Part 1. Identify the part of a phospholipid molecule labeled A in figure 1.1 that forms bonds with the phosphate heads and with the fatty acid tails. So guys, we have to identify this part A of the phospholipid. So this is the phosphate group and these are the fatty acid tails. So basically phosphate group and fatty acid tails are attached to glycerol. So A is glycerol, which is attached to the phosphate heads and with the fatty acid tails. So the answer is glycerol. Alright, let's move to the next part, part 2. Cholesterol is an important lipid component of many cell surface membranes. Figure 1.2 shows the structure of a cholesterol molecule. So guys, in front of us is the cholesterol molecule in figure 1.2. We can see that cholesterol molecule has a polar hydroxyl group and a non-polar part, right? Alright. Using the information in figure 1.2, explain the orientation, position of cholesterol molecules in the phospholipid bilayer as shown in figure 1.1. So guys, we can see the orientation of cholesterol molecule in the phospholipid bilayer. This is the cholesterol molecule. We can see that the OH group, which is the polar part of the cholesterol molecule, is in contact with the phosphate heads because the OH group is hydrophilic as well as the phosphate heads, so they fit together. Whereas the non-polar part of the cholesterol is in contact with the fatty acids, which are hydrophobic, right? So because this part of the cholesterol is hydrophobic or non-polar, so it is fitting with the fatty acid tails, which are also hydrophobic or non-polar. So we will write that the OH group, the OH, or we can say the polar hydroxyl group, fits among phosphate heads which are polar or hydrophilic, and non-polar part of cholesterol molecule fits in between hydrophobic fatty acid tails. Hydrophobic fatty acid tails. Alright. State one role of cholesterol in phospholipid bilayers. So guys, we all know that the role of cholesterol in the cell surface membrane or phospholipid bilayer is that cholesterol maintains the fluidity of the membrane. So cholesterol maintains or regulates the fluidity of the membrane. Cholesterol prevents the membrane from becoming too fluid or too rigid. B part 1. Explain why sodium ions cannot cross phospholipid bilayers by simple diffusion. So guys, over here they are asking that why sodium ions cannot flow through the phospholipid bilayers by simple diffusion. That's because sodium ions are charged, and you all know that charged particles are repelled by the hydrophobic tails or the hydrophobic fatty acid tails in the phospholipid bilayer. That's why ions cannot flow by simple diffusion. So we can mention over here that sodium ions are positively charged. Sodium ions are positively charged. Sodium ions are positively charged, and you will write that hence, yeah, therefore sodium ions are repelled by hydrophobic fatty acid chains. hydrophobic fatty acid hydrocarbon chains in the phospholipid bilayer. So they cannot pass by simple diffusion. You should remember that simple diffusion is the diffusion of particles from a higher concentration to lower concentration through the gas between the phospholipids, right? This is simple diffusion. Let's move on to the next part, that is part 2. Ions and some molecules move across cell surface membrane by facilitated diffusion and active transport. Compare the facilitated diffusion and active transport by stating one way in which they are similar and two ways in which facilitated diffusion is different from active transport. So guys, you should know that active transport always occurs through a carrier protein. For active transport, you always require a carrier protein, right? Whereas if we talk about facilitated diffusion, so facilitated diffusion can occur through a channel protein or a carrier protein. So one similarity is that both the facilitated diffusion and active transport, they both occur through a carrier protein, right? So we can write in a similarity that both facilitated diffusion and active transport, they require a carrier protein. This is the similarity. What about differences? Let's just write the similarity first, that both facilitated diffusion and active transport require a carrier transport protein, both facilitated diffusion and active transport require a carrier protein in the cell surface membrane. Right? Another similarity is that both the facilitated diffusion and active transport, both are of specific molecules. What is meant by this statement? That because facilitated diffusion is brought about by a transport protein, and same goes for the active transport. So you know that transport proteins are specific and they only transport specific molecules. So you can say that both facilitated diffusion and active transport involves the transport of specific molecules. This is also one similarity. What about differences? You should know that facilitated diffusion involves movement of molecules slash ions from a higher concentration to lower concentration. However, this is vice versa for active transport, whereas active transport involves movement of molecules slash ions against the concentration gradient, that is from lower concentration to higher concentration. All right. What is another difference? Another difference is that facilitated diffusion is passive, right? Facilitated diffusion is a passive process. It does not require ATP from respiration. So you can say facilitated diffusion is a passive process, does not require ATP, right? Whereas active transport requires ATP. So guys, these are the differences, that active transport requires ATP, whereas facilitated diffusion is a passive process, it does not require ATP. Facilitated diffusion involves the movement of molecules or ions from a higher concentration to a lower concentration, whereas active transport involves the movement of molecules or ions from a lower concentration to higher concentration. If we talk about similarity, then both facilitated diffusion and active transport, they require a carrier protein in the cell surface membrane. Let's move on to part C. Prostaglandins are small lipids produced in many tissues of the body. One role of prostaglandins is to cause inflammation at the site of an injury or infection. Inflammation is the normal first response of the immune system to injury or infection. Cyclooxygenase or COX is an enzyme that catalyzes one of the steps in the reaction pathway for the formation of prostaglandins from phospholipids. The reaction pathway occurs in the smooth endoplasmic reticulum of cells. Part of the reaction pathway is shown in figure 1.3. So guys, in this part, they're saying that there are some molecules known as prostaglandins. These are lipids, and these are synthesized in the smooth endoplasmic reticulum from phospholipids by the action of COX. And they are saying that prostaglandins cause inflammation at the site of the injury, and inflammation is basically the response of the immune system to the injury or infection. So part 1 says that suggest an advantage for this reaction pathway occurring in the smooth endoplasmic reticulum of a cell rather than in the cytoplasm. So they are saying that what is the advantage of this pathway occurring in the smooth endoplasmic reticulum rather than the cytoplasm of the cell. So what could be an advantage? We all know that smooth endoplasmic reticulum is involved in the lipid synthesis, right? So that's why prostaglandins are being made, or this reaction pathway is going on in the smooth endoplasmic reticulum, because this pathway involves lipid synthesis. Prostaglandins are lipids. So you can say that smooth endoplasmic reticulum synthesizes lipids, right? So this reaction pathway should occur in the smooth endoplasmic reticulum. You can also say that smooth endoplasmic reticulum, because it synthesizes lipids, it has phospholipids, right? Or you can also say that smooth endoplasmic reticulum membrane has phospholipids, which can be used as substrates for this pathway, right? Phospholipids in the membrane of the smooth endoplasmic reticulum can generate arachidonic acid, and then arachidonic acid is the substrate for COX, which eventually makes prostaglandins. So you can write over here that smooth endoplasmic reticulum synthesizes lipids, and prostaglandins are lipids. You will also write that membrane of smooth endoplasmic reticulum provides phospholipids, slash arachidonic acid as a substrate for COX. All right. Part 2. Sometimes inflammation can have side effects such as pain. Aspirin is a drug that can be used to reduce these side effects. Aspirin reduces the catalytic activity of COX enzyme by modifying the R group of one of the amino acids. Suggest how modifying the R group of an amino acid in the COX enzyme can reduce the catalytic activity of the enzyme. So they are saying that aspirin is a drug which can reduce the catalytic activity of COX enzyme. And aspirin does this by modifying the R group of one of the amino acids. So they are asking that if R group of one of the amino acids in COX is modified, so how will this affect the catalytic activity? Or how can this reduce the catalytic activity? So simply, we know that if the R group is modified, then the bonds holding the COX molecule in the tertiary structure will change. So modifying the R group can change the bondings like hydrophobic interactions, hydrogen bonds, disulfide bonds, and ionic bonds. When these bonds change, what happens? The shape of the tertiary structure changes, right? The tertiary structure changes, the shape of the active site changes, substrate is unable to fit, right? And this is how catalytic activity is reduced. So we will write over here that modification of R group, modification of R group, modification of R group will cause the bonds such as hydrophobic interactions slash hydrogen bonds slash ionic bonds or hydro or disulfide bonds to break, right? We can also mention that modification of R group will cause the bonds such as hydrophobic interactions, hydrogen bonds, ionic bonds, or disulfide bonds holding up the tertiary structure, holding up the tertiary structure to break or change, to break or change. The shape of the active site changes, the shape of the active site of COX changes. COX is the enzyme that is cyclooxygenase. So the shape of the active site of COX changes. Substrate is unable to fit. Substrate is unable to fit, right? Let's move on to part 3. Prostaglandins are examples of cell signaling molecules. Outline the process of cell signaling that leads to a response by cells involved in the inflammation. So guys, over here we just have to outline the process of cell signaling. So this is a very basic question in which you have to outline the process of cell signaling. So how does the cell signaling occurs? You will simply say that in a cell signaling event, signaling molecule is released or secreted by signaling molecule. For example, over here, prostaglandins are secreted by signaling molecules are secreted by cells which travel. Signaling molecules are secreted by cells. Signaling molecules travel towards and bind to the receptors on the target cells. You all know that in cell signaling what happens, there are some cells that are secreting the signaling molecules, and then there are target cells which will receive these signaling molecules. For example, if I have to give you some message, so I am conveying the message and you are receiving it. Just like that, signaling molecule is a message which is released by some cells, and this message is then received by the target cells, right? So signaling molecules travel towards and bind to the receptors on the target cells. What happens next? The receptor changes shape. Which results in the formation of a second messenger, Then second messenger activates an enzyme cascade. And because of the activation of enzyme, responses brought about by the cell. So we are done with question number 1. Now we will move on to question number